Welcome once again to this channel for NEET Biology, and today for class 11 special chapter small video for the anatomy of plant anatomy, the vascular bundle, the different types of vascular bundle. After the completion of this chapter, we will go through all the questions of NEET from this part, that is the vascular bundle. What are the I have taken the pictures, diagrams and different informations for your NEET exams that are essential. So please go through this video up to the end and you can answer any question from this part. So let's start, and today we'll discuss what are the difference between the monocots and dicot stems and roots. What are the different functions of the parts of the anatomical parts, different cell layers like endodermis, why they are called starch strips, why they are called suberized layer, what are the function of pericycle, what are the endodermal layer, what are the pericycle, what are the function of protoxylem, metaxylem, what are the function of what is actually the endarch and exarch, why it is called exarch and endarch, from where the secondary growth initiates, from where the medullary rays develop. So all we will discuss here. So go through this, and this is very useful video for you. So let's start. There's a types of vascular bundles. Vascular bundles are, there's a xylem and phloem, but they are present in different manner in different plants. Example here with a radial pattern where xylem and phloem are not present together. Xylem present separately and phloem present separately and in an alternate fashion. Here the xylem, here the phloem, here the xylem, here the phloem. So if we see that the roots of both dicot and monocot, you will see that here the root anatomy, and you will see easily that there is a xylem, here the phloem. There is a phloem, there is a xylem, phloem, and repeat. So this is a typical example of a radial pattern. It's a dicot root. Not only the dicot root, the dicot as well as monocot root, both are of radially arranged vascular bundle. What happened with this? The protoxylem is exarched, that is the protoxylem form first towards the periphery and metaxylem towards the central region. So this one is most important. There's an exarched vascular bundle we will see here. Exarched vascular bundle, and all the traits are the same. That is the endodermis, the pericycle. The endodermis, pericycle is present beneath the endodermis. Endodermal layer is present. That's the suberized and impermeable, highly impermeable layer. That is the endodermis. It is actually inner layer of the cortex. So we'll discuss later in details. So protoxylem and metaxylem is formed here. So in this case it is important, radial type present alternately. And you will see that the conjoined closed type. This is a monocot special. The monocot stem. In case of monocot stem, the vascular bundle is present in a in conjoined and closed condition. Conjoined and closed means they are present in a scattered manner. Vascular in this case, in case of monocot, the vascular bundles are present in a scattered manner. That is here the patch like, not radially arranged like that of in case of dicot stem. In this case, vascular bundles are present in scattered manner. So what happened in this case? In between phloem and xylem. Xylem and phloem not alternately present here present together. But one important thing is that no cambium is present. No cambium is present, so no secondary growth is there in case of monocot stem. So the monocot stem, xylem and phloem is present in a patch in separate, present together, but in a discrete manner, in a discrete manner, not in a radially arranged manner. So this one is most important. In this case, here you see that this is a xylem and these are the phloem. This is a xylem, these are the phloem, and outside the phloem there is a bundle set, highly specialized scleroderma cells, bundle set cells is present. So parenchymatous cells present is the phloem and xylem present together. No cambium. You'll see that no cambium is present. So no question of secondary growth. If cambium is present, not present, no secondary growth is there. So it is very important. So let's start with the conjoint open type vascular bundle. Conjoint open, that is the conjoint, the xylem and phloem present together, but in between them the cambium is present. Cambium, we know that cambium is a specialized structure, specialized tissue system which produce continuously secondary xylem or xylem towards the inner side and phloem towards the outer side. That is called vascular cambium or fascicular cambium. So secondary, during the formation of secondary growth or when the plants increased in size or girth, so during that time, in case of Dicot stem, we know the Dicot stem are larger and they are the diameter is very high. So this is actually due to the wood, and secondary xylem, the secondary xylem produced by the cambium is a huge amount. So this huge amount of secondary xylem are produced by the cambium. So this is a cambium is present in case of Dicot stem. In this case, cambium produces secondary phloem outer side and secondary xylem toward the inner side. It's called wood. Wood is nothing but a secondary xylem. So in this case, here the tissue system, in this case tissue system, phloem is present outside and xylem is present here. In between them, cambium is present. This is a parenchymatous tissue, specialized and produce huge amount of xylem inside and phloem outer side. So this is a cambium, and this xylem, if you carefully note that this xylem is opposite than that of the xylem present on a root. Here the smaller, that is the protoxylems are present towards the central region, but if we see that in case of here the protoxylems, that is the smaller xylems, firstly formed xylems are present towards the periphery, not towards the central region. And so this is the opposite phenomenon, and this one is most important phenomenon, that is xylem is in exarch here, but in case of stem, xylem is endarch. So vascular bundle is endarch. So protoxylem present inside, not outer side. This one is most important. So all three types of vascular bundle is there: the radial type, conjoint closed type, and conjoint open type. Conjoint closed type and conjoint open type. So this one is most important, and most important thing another is how we will stain the vascular bundles. So there is a two type of staining system. There is the safranin and methyl green or light green. So safranin stains the lignins and dead tissues like xylems, red orange in color, and the tissues contain lignin, suberin containing dead tissues are stained by the safranin. In case of methyl green especially stains the cellulose containing phloem, pith, and epidermis or living cells. So methyl green and safranin, this you have to understand and remember that for what the safranin and for what the methyl green, these two stain systems are produced. To separate them, here you will see that there is the red colored, that is the lignin containing, suberin containing, and the dead tissues, that the xylem, are actually stained by the red colored stain or orange color stain, safranin, and blue green color of stain, that the living cells taken the blue green color. It's a phloem, some part of phloem in phloem parenchymatous are living, and parenchymatous, all parenchymatous cells are blue green color due to this stain. So let's start the Dicot stem discuss. What is actually the, if we cut a section, transverse section, what we will see from outside, the outermost layer is called epidermis. Epidermis, the parenchymatous living structure. Then the hypodermis is hypodermis, in case of Dicot stem, hypodermis is cholenchymatous. We know that the cholenchyma is unequally thickened living cells. So hypodermis is a cholenchymatous. Then hypodermis is a part of a cortex, cholenchymatous, and then the cortical layer. This cortical layer is the parenchymatous tissue, and as well as not only the parenchymatous, they are living also. Not only the parenchymatous, they are living and they have an intercellular space, conspicuous intercellular space between them. So This is the most important, that is the parenchyma testis, cortex. And just below the cortex ended with a specialized wall is called endodermis. So endodermis is the most important layer. In that sense, it is actually end or boundary of your cortex. And after the endodermis, there is another layer, it's called pericycle. Pericycle is the first layer of the vascular bundle. So the last layer of the cortex is endodermis, and first layer of vascular bundle is the pericycle. So just below the pericycle is the phloem. That's the phloem produced by the cambium, and in this case the vascular bundles are present radially arranged, and in between two vascular bundles there is a parenchymatous tissues which is called as medullary rays. These are medullary rays are produced by this fascicular cambium, this fascicular vascular cambium. So this one is very important. So phloem is there, and inside the phloem then the cambium, then after the cambium xylem is there, and ultimately at the central region the parenchymatous with intercellular spaces pith are there. So they are all living. Here the xylem is not living. So this one is most important. So this is a chronological your steps: epidermis, hypodermis, cortex, and endodermis, pericycle. You have to remember it somewhat by seeing these structures. So phloem, cambium, xylem, pith. So and this is important. Epidermis. So this is the tissue system. Here the epidermis, there is the cholenchymatous hypodermis, then parenchymatous cortex, and then endodermis, and this is a pericycle. This is the endodermis. Endodermal cells are a specialized structure. We will discuss later. That's the endodermal cells are a specialized barrel-shaped structure. Endodermis also contains amyloblast. So why they call starch sheath also, and endodermis is actually formed impervious layer between the cortex and vascular bundle. Why? Because endodermal cells contain Casparian strips that are specially formed by the suberin, and suberin later on at the maturity, the suberin and lignin together form a impervious or totally waxy impervious material that prevent the apoplastic movement of the water from the cortex to the central region. So from central region to the cortex through apoplast, it is a main hindrance. It is a main of the hindrance, that is the no exit of water and hormones or minerals from here to here. Only passageway is present in some cases. Passageway are actually present in case of roots, where some cells of the endodermis allow the entry and exit of the water and minerals through the muscular bundles. So how the water and mineral substances are passing from the central region to the outer side? It is the medullary rays. So medullary rays are the most important functions. Have a most important function. So later we will discuss here. Here the phloem region, and this is a pericycle, and this is a phloem region, and this is a cambium. This is a xylem, and it is a endarched xylem. Protoxylem is present towards the center. Then they will see that the monocotyl stem. Here the monocotyl stem, the vascular bundles are not arranged readily, but they are scattered, and we have said the closed, and there is a closed conjoined type, not open type. So no cambium is present, vascular bundle is present, xylem, phloem, and ground tissue are there. Here the intercellular spaces, huge intercellular spaces are there. So hypodermis is present. Hypodermis is cholenchymatous hypodermis here, not cholenchymatous. As in case of dicots, we have seen that there is a cholenchymatous, but in this case it is sclerenchymatous. However, vascular bundles are scattered, surrounded by sclerenchymatous bundle set. This one is most important trait. We will see later in a C4 plant, where we will see that it is this one is a surrounding layer is there. It's called vascular bundle or bundle set. So bundle set cells are sclerenchymatous. Phloem parenchyma is absent, absolutely absent. This one is important hallmark trait of monocot phloem. That is, phloem parenchyma is absent. Now we'll see that the root structure. You will see that the same root structure in monocot and dicot. same root structure. Here the first epidermis, cortex, epidermis, cortex, endodermis, pericycle, phloem, and the phloem is of exarch type. That is the protoxylem present outside outer region and metaxylem with the central region pith, an alternative radial type of arrangement of vascular bundle. Protoxylem is exarch, radial vascular bundle, vascular is surrounded by endodermis, no cambium is present. So this is the overall structure of vascular bundles, vasculature, and we will see that the endodermal structure. Endodermis is the innermost layer of the cortex and inhibit the apoplastic movement through the cell wall. So apoplast is most important phenomenon where we see that the movement of the water molecules occur through the cell wall. So this is the main hindrance where from cortical region to the central region or the steely region, no water molecule or mineral molecule will transfer through apoplastic way due to this endodermis layer. Why endodermis contain the waxy? I have mentioned that the suberin containing substances and that called impervious layer. So it is a boundary between vascular and steely also. This one is important. Involved in peripheral perception of gravity. Gravity perception is most important. It stores the myeloplastic starch sheet. So it is important in pericycle. Pericycle is the most important thing, is the outermost layer of the vascular bundle or steely that lies just inside the endodermis, as well as it is the scleral cavities and present in both roots and shoots. Medullary rays. Medullary rays are present in between the vascular bundles. Parenchymatous, they are formed by vascular and vesicular cambium, important for the transport of minerals and water to the periphery. So these are the overall structures as I mentioned, and if we have any query you can ask in a comment section and so we can answer it. So you have to read it in that way. Thank you very much for giving time while